Recording. Good afternoon, guys. I'm Deepak from Center for Neuroscience. Let's get started with the tutorial for week 3 lectures on the course Demystifying the Brain. First, let's discuss about how the visual processing, through the visual processing, we'll understand how the physical signals are processed by your brain to make sense of the real world. So, for starting with visual processing, let's start with the gateway to the visual processing, that is the retina. So let's say you're looking at an object like this cup. The light from this cup falls on your lens and then get focused on your retina. So now how the retina is structured. So the light falls on this pigment epithelium. So when light falls, there are these light sensitive cells. This acts like a transducer for converting the light signal to electrical impulses, and then further processing takes place in your brain. So in the retina has three types of layers. In first layer it has transducers which contain cells called rods and cones. These are receptors which act like an entrance for converting the light signal to electrical impulses. Then there is a layer of bipolar cells. This is this acts like helps in processing further, and then you have an important layer called retinal ganglion cells, which actually the outputs of this form the optic nerve. So you see here the yellow color here. So this forms the optic nerve. All these fibers from this retinal ganglion cells combine and go and form the optic nerve. And at the center of optic nerve you see a gap, and this is called blind spot. Okay, let's see how the visual processing takes place now after seeing the anatomy. First, getting to the receptors, the rods and cones. So here you see a picture of eye, and you see retina here, you see some angles. So this angle, it is marked as 0 degree. This is called fovea. This is where the light gets focused on to wherever you're fixating. Like if you're looking at some point, the center of that point uh, projects onto this fovea, and the distribution of these receptors are different on the retina. So here you see a graph where the x-axis shows the degree from deviation from fovea, and this shows the density of the types of cell, either rods or cones. So you see at the fovea you have high density of cones. Fovea has high density of cones, whereas low density towards the periphery. On the other hand, rods have high density towards the periphery and low, almost, zero density at the fovea. So these two different types of cells have different properties that helps in perceiving the visual world differently, in different situations, not differently. So those both are involved in all the time in your vision, but they have different properties. So coming to cones, the the main advantage of cones is it helps in high visual acuity. whereas rods help in rods have high sensitivity. How is that implemented in the brain? So um, let's take rods on one side. So the thing with rods is they have convergent connections. So they all converge onto a downstream neuron here. On the other hand, you take cones. Cones have point-to-point type connections where there is a single downstream neuron for each cone. Yeah. So coming to sensitivity of rods. So here, because it has summation type convergent connections, small inputs here will just sum up and reach the threshold, and then the downstream neuron fires and the further processing of information takes place. Whereas if you take cones, if there is a small input, the downstream neurons didn't reach their threshold, the input current is still small amount and the downstream neurons doesn't reach, and there is no output signal for further processing. This way there is high sensitivity for rods, but coming to cones, why there is a high visual acuity. Let's say you have good amount of input coming from 2 points, two neighboring points in space in actual visual world. Then what happens is these two spaces will just these 2 points, neighboring points, have independent processing in the downstream areas, and therefore the downstream areas can still differentiate the two neighboring points. But if you take two neighboring points on in the case of rods, since it gets convergent summation, the signals get mixed up and the downstream neuron is not able to differentiate between the two neighboring points. This way, cones have higher acuity and rods have higher sensitivity. And during night vision, since the amount of light entering your eye is less, the high sensitive cone takes the processing, gets the processing advantage and are more active during the night. Rods are more active during the night because of their high sensitive capability. This is about receptors. Let's see how the further processing takes place. So before that, let's talk about a term called receptive fields, which is a very important term in neuroscience. So if you take receptive fields, let's define it first. Where is it and of what? So we generally say receptive fields of a neuron. So it is defined for a particular neuron of your interest. Neuron of interest. Okay. So we generally ask what is the receptive field of a particular neuron. But where is it located? It is located on the receptors. This can be defined for different sensory systems, either for vision or touch senses, different types of senses, auditory, etc. So, receptive field is an area on the receptors. Receptive field of a neuron is the area on the receptors for which this selected neuron activity gets modulated. So, if you make some changes in the area of receptors, there is a change in this particular neuron. So the changes in this receptive field for which this neuron responds is the receptive field. So for example, you take this plane, let's take an example of light, and you throw spots of light here on the area A, represented by area A. You see that there is some activity, but this is a spontaneous activity. This is like a baseline activity which is always present for a given neuron. Like it's some random activity going on, but there is no there is no particular modulation of this neuron due to this light spots thrown on the area A. But When you throw a light on area B, the center area B, you see that as soon as it starts, like whenever it is on, you see the firing rate increases and it is there as the light is present. And when you throw a light onto area C, you see there is no firing, it is zero, like even the baseline is not there, and then once you turn it off, the firing starts. So So this area, that is area B plus C, forms the receptive field of particular neuron for which you are measuring the signals. So these signals are from a neuron of your interest, and this area B plus C forms the receptive field of this neuron. And this area is defined on the receptors. For example, like you can define a receptive field for a neuron connected to the rods or cones, something like that. And yes. So this forms a receptive field. So you see like this kind of receptive field where you have excitation at the center and inhibition at the surround, or inhibition at the center, excitation at the surround. These types of the receptive field are called center surround receptive fields. Center surround receptive fields. You can have excitatory center, inhibitory surround, or inhibitory center, excitatory surround. So like it depends on the neuron that you're measuring and the neuron properties or how it is structured. So this is about receptive fields, and in the retina we saw that there are these cells called retinal ganglion cells. So these cells have a center surround receptive fields. So different cells map onto different space, different point in the space, and therefore like each cell has a center surround receptive field. It was like seen in the experiment. Then coming to next levels of visual processing, actually in the cortex. So we have discussed retina. Now let's go into the brain. So here I'm showing a visual pathway, complete visual pathway from the light entering your eye to the processing in the brain, especially in the primary visual cortex. So uh, how is a visual system structured is it has a bilateral structure. What I mean by bilateral structure is you have a right hemifield, left hemifield. This is a whole left hemifield falling onto this eye, like whole left hemifield falling onto this eye, and this part falling onto this eye. So you have both eyes getting input from left hemifield of the visual space. Hemifield in the sense you have left and right. So left visual field, right visual field. From left, both the eyes gets input, and the neurons, the part of retina that is responding to left visual field have this optic nerve coming here, and at the optic chiasma you know they just go on one side of the cortex. So this is called one hemisphere of the brain, this is called other hemisphere, like left hemisphere and right hemisphere of the brain. So left visual field is being projected onto right hemisphere, and here you have a very important part called thalamus, and the main nucleus in the thalamus that is involved in visual processing is called lateral geniculate nucleus. So the left visual space falls on this one from two eyes. It projects onto lateral geniculate nucleus, and from there it goes into the primary visual cortex here. From here, like it goes into the like the brain processes the information. Uh, this is a primary visual cortex. It is also called as V1. This is how visual path is organized. Now let's see how the receptive fields and different properties of lateral geniculate nucleus are structured. So through experiments, they saw that LGN also have similar to uh, retinal ganglion cells. They have a center surround receptive fields, as you see here. So center spot you have high firing, you fill the spot you have still higher firing. Once the light extends outside the excitatory area, slowly the firing rates decreases, and when it fills, it is like both are getting utilized and you have a baseline activity. So it's the same way. LGN also has a similar receptive field structure as that of retinal ganglion cells, and this nucleus LGN in thalamus is a bilaterally symmetric structure. It's a like it has both left and right, and it's symmetric. And if you see a part of thalamus here, you see a laminar structure. What this different layers mean in this LGN is it has neurons responding. The blue color represents contralateral eye. I'll tell you what is meant by this ipsilateral eye. So what is meant by contra and ipsi? So you have LGN receiving inputs from two eyes, like right eye, left eye, from like let's say one visual field here, that is left visual field, and LGN receiving inputs. So if you're measuring from right hemisphere in the brain to the right hemisphere, the eye on the right side is called the ipsilateral eye. Ipsilateral is same. Contralateral is opposite. So from the opposite eye also you get inputs. So these inputs are organized in the layered fashion here, where each layer has inputs from different eye. So this is a way how it is structured, and there are 6 layers in this LGN, and this is about the laminar organization of the LGN and how the receptive fields are structured. Now let's go to another concept called retinotopic maps. So, uh, so inputs from real world go into your brain, but uh, so certain points have correspondence in your brain, like certain point in the actual real world points to a place in your brain. So this correspondence, like how the real world space is present in the brain at particular point of interest, you call it as a like mapping between real world and the space in your brain, like how which neuron responds to which point in the real space. So this kind of correspondence mapping is called retinotropic maps. So for example, let's say you're looking at 3 points on this cup. You have A, B, C points which projects onto your retina like this, A, B, C, and each point goes to this LGN neuron A, B, and C. This forms like this correspondence, the point on your retina and the point on your this correspondence, this mapping is called retinotopic maps. So when you pass an electrode in one particular layer, let's say this layer 6, you have 3 points corresponding to actual space on the retina. So this A corresponds to A, this B corresponds to B prime corresponds to B, and C prime corresponds to C. This correspondence where neighboring points are still neighboring in your LGN, this layer, it is also same in other layers. So this type of mapping is called retinotropic mapping. Now if you take a perpendicular track, all the neurons on this responds to the point at this same C position. So through experiments they found this is how the retinotopic map of LGN is structured. Now let's go into the brain where From LGN, the further processing takes place at primary visual cortex. This is primary visual cortex, which is also called V1. So how is the receptive fields of V1 structured? So V1 have a receptive fields like this. You see in figure A here. You have excitatory field, excitatory ellipse here, followed by inhibitory elliptical field here. So whenever a spot of light or a bar is present here, it just gets excited, as you see, and as it has orientation changes, it slowly covers the inhibitory area also, and therefore it's as the orientation slowly changes to horizontal, it just uh, goes like this. The firing just changes. So the neurons in visual cortex have orientation selectivity. So what I mean by that is different neurons in visual cortex have different types of similarly structured receptive fields. What I mean, so here you see this neuron is having this type of structure. You have other neurons having some oblique structure like this. You have certain neurons having completely horizontal structure. So each neuron has some orientation specificity because of this. Like if you take a neuron with minus 45 degree uh, selectivity, you see that neuron's receptive field is structured like this. And if you see like the 0 degree receptive field, it is like this. So each cell in visual cortex have some orientation selectivity, and you can plot for every neuron a curve called tuning curve, which tells about its orientation selectivity. So this is called tuning curve. Tuning curve. So you for this particular neuron, you have highest firing rate for vertical orientation, and this tells this neuron has a vertical selectivity, and has the as you rotate the bar, the firing rate slowly decreases. So this is impulse per second. This is like spikes per second, like number of spikes per 1 second. So this way you can have different tuning curves. You can, like, if you take some neurons, you'll have tuning curves like this, like this, like this. Some neurons respond to 0 degree, and at 45 it just goes 0. And similarly, this responds to some uh, 50 degree, and you can have this kind of tuning curves, and further downstream it has processing for this. So responding to this is like responding to some edges of light, or like in your real world you have edges of an object at different orientations, and it helps to represent the edges. These cells, the cells in V1, helps you to, like, there are more complex cells, and these are also called simple cells. The cells in V1 which have this kind of, where you can come up with a concrete receptive field, are called simple cells, and they have this receptive fields. There are also other types of cells in V1 called complex cells, and these complex cells have different types of receptive fields, which we can't make a concrete receptive field, but they have convergent inputs from different simple cells, and therefore they respond to complex features of the visual object. For example, you see this neuron which responds to moving orientations, and it has a moving orientations selectivity for moving upward at 45 degree stimulus, this light bar moving upward. This has the high selectivity for this particular neuron. And there are other neurons which respond to corners and only having particular, lengths. These are called end stopping cells. So you have, if you just cross particular length, and it just stops firing. If you're inside, it is low firing. If you're exact, it has high firing rate. So these are called end-stopping cells. These have slightly complex receptive fields, and you cannot particularly come up. For example, if you see a simple cell, we have seen how the receptive field is, but for a complex cell, it has a weighted sum of these different receptive fields. You have like some W1, W2, W3, W4, W1 times receptive field 1, some kind of linear combination of these receptive fields, and then you have this like, so for this you can't particularly draw a receptive field as we have seen in this V1 simple cells. So this is about the primary visual cortex. Uh, Coming to retinotopic maps in V1 and some terms like cortical magnification. So even in V1, if you pass a electrode like this across the surface here, you see that each point has a receptive field something like this. So neighboring points corresponding to neighboring receptive fields on retina. So this way you have correspondence maintaining between actual visual space, neighboring points in visual space correspond to neighboring points in maps, and that way you maintain the topological structure, and that is why these are called maps. So in real world also the maps maintain the topological structure, like point corresponding to space have neighboring points in real space have neighboring points in the map also. So you see, let's see, this is a real like visual field. This is a in real world. Let's just draw some concentric circles in the real world, and you see this has a fixation point where you are fixating your gaze, and the closer points are mapped to further away and have greater cortical space representing that area. As you go far away from the center, you see the map a- the amount of area that processes this part reduces. So this is called cortical magnification. The points closer takes this larger. So this kind of thing is called cortical magnification, and one can calculate what is the factor of this, like how much is this magnification factor, like how much this point is magnified in the cortical space. So why do we need more area for this is because when you're foveating you want like many details from the area where you are fixating. So that way, like with higher cortical space, there are higher processing resources allocated to that particular space in the visual field. So this is called cortical magnification, and this is how the receptive fields are still present in V1. So you see here in the visual field where you're fixating here, you see that gets magnified here in the cortex. So this is the cortical magnification, and you have retinotopic maps, and with uh, these graph you can see there are few experiments performed where you give a radioactive safe and radioactive glucose to the neurons, and whenever they fire they just make marks like this depending on where you light up in the actual visual space. So this is actual visual space, and this is the cortical space in your cortex, basically in your visual primary visual cortex. So you see these patterns, like wherever you light you have corresponding points. Like if you take point H here, it has a point in cortex, corresponding point in cortex, and you take point F, you have corresponding point in cortex. And so as we have discussed, these are eccentricity, like particular eccentricity like 1 to 3. So this is a distance from the center. So this one has like 1 degree, and this has 2.3 degrees, something like that. So these are called degrees of visual angle. We measure because this degrees of visual angle considers the distance of you also. Like this metric, this particular metric is actually a tan inverse of, you know, uh, so for example, you are fixating here, the object is here, and you are here, this distance d and this distance a. You can say in distance also, but that doesn't account for how far you are from the object. So in order to account in a combined fashion, there is a metric which combines these two, that is this theta. Theta is nothing but tan inverse d. So this this way, you like using this degrees of visual angle, you can account for both the distance of you from the object as well as the distance of the object from the center. So that is why we measure in degrees of visual angle. Um, so the relation between the corresponding points in this is given by a a mathematical equation, that is the cortical location is some constant lambda times natural logarithm of a small z here. So what the small z mean is eccentricity by some constant epsilon naught. This represents eccentricity times some complex exponential of this angle A. What is angle A is, this is a azimuth. So we have discussed eccentricity is distance from the center. So let's say you have a actual visual space, you are here. So this is, sorry, this is degrees of visual angle, this is epsilon naught, this angle is epsilon naught, and if you rotate in this visual space, this angle is called azimuth. This is the angle A, and this way you can have a corresponding points in cortex from the real space. So this is how the retinotropic maps of primary visual cortex is structured. And as we discussed, when you go perpendicularly, you see that all have overlapping receptive field. So this is like going through the column, going perpendicularly to the cortical surface, and each column here have a particular orientation selectivity. We have discussed like each cell has some orientation selectivity. Similarly, when you go perpendicularly, you have all the cells responding to particular orientation, and when you move obliquely across the surface, you see the orientation selectivity changes. This has one particular orientation selectivity, this has another orientation selectivity, and each point corresponds to different point in the real space, depends on the retinotopic map. So this have orientation selective columns, these are called orientation selective columns. And now you have similar to like you see layers coming from different eyes on the LGN. You have similarly here also, like alternating between left and right eye. So this gets input from left eye, this gets input from right eye, and you have different layers again, left eye, right eye. This way the columns are there, and each column has particular orientation selectivity. So these columns are called orientation selective columns, and these columns are called ocular dominance columns. Like from so this the cells in this area receive input from both eyes, but dominantly from left eye and dominantly from right eye. That way the naming has come, that is ocular dominance, which eye is getting dominated space in that particular column. So since you have a space with both orientation selectivity and ocular dominance, you get some map like this depending on which orientation selectivity it has, and you have some columns with different ocular dominance, like left eye, right eye, again left eye, something like this, right eye. This way the visual cortex is structured, And you see that as you go across here in this schematic, the orientation selectivity goes in a linear, like nice fashion, incremental fashion, but this is not true for all the area, the whole area in visual cortex. So different areas have, if you take this particular thing has incremental orientation selectivity like this, but if you see this, it has a circular orientation selectivity. So there are different points associated with how the fields, like different points in the primary visual cortex have different names and terms. And this is how the columns of V1 are present, which has orientation selectivity and ocular dominance columns. And this color maps gives a big picture of how the ocular dominant and orientation selectivity is structured in the visual cortex. This is about V1, and these maps you see here we only discussed for sense visual senses, but it is present for different kinds of senses. You have retinotopic maps for auditory cortex, for somatosensory cortex, for like your touch senses, etc. So these maps are very important in the brain so that you know like which area, like you have to maintain the topological structure of the real world, and because you have to make sense of real world, like neighboring points in real world should correspond to neighboring points in the cortex. Yeah, neighboring points should have neighboring areas responding to each other so that they still have intact topological structure. So basically the main aim of map is to maintain this topological structure. So these are the two main principles of map. So proper neighbors should correspond to proper neighbors, and also the context, and closely related information is kept close together. For example, in visual space you saw neighboring points corresponds to neighboring points in the visual cortex, but for sound the information is actually encoded in terms of, like, it makes sense to encode in terms of frequency. Therefore same similar frequencies are having neighboring points in the auditory cortex, and they have like frequency type retinotopic maps, not the spatial maps, rather it has frequency maps. So how are these maps like able to evolve and get structured for the world you are seeing? So it is basically a high dimensional feature, and you have like the point in real world is high dimensional, but this is like in cortical sheet you have particular point corresponding to that particular point, and this is like two-dimensional map. So you're basically reducing the real world parameters to certain map. Let's say you have an input features vector, like it has multiple features that your brain wants to represent as a map. You have different components x1, x2, so on. Let's say xn. So here, if you take one point x, like each component, you have a vector x here, and this this x actually can connect to every neuron in this structure. This is, let's say this is a map structure, which you have to, it has to evolve and form a map. So this is the initial map you have. So here every component, let's say you have x1, x2, x3, every component is connected to every other node, every node here. So you have all this xn components also coming to one single node. Let's take this is the node of our interest. you have all the vectors projecting onto this. So each line here has a weight. So this is 1. So W1 and W3 and W2 and W1 and you have like this. So you have X input vector and you have corresponding weights coming to particular node. Like that you have for every node. Every node you have like different weights coming from that input. So these maps are best when the distance between like the Euclidean distance between the X and the weights W, like x1 to xn is this vector, and you have correspondingly for each node you have a weight vector w1, and for one node it is this, sorry, uh, wn1, wn2, and so on, wnn have this weight vector. And this map is formed such a way that the input space and this weights the distance between these two vectors, like you have this vector x bar and you have a weight vector for corresponding to particular node, here node 1, the distance between this is reduced, so that way it optimizes the map. And you want to maintain a neighboring structure, so you have that constraint. How you can provide that constraint to this particular map algorithm is you just like you find a weight of this, like optimal activation optimal map of this, and you just like you also interpolate that to neighboring neurons such that the neighboring context is still maintained. And like basically you have these two constraints, and that is possible through minimizing the distance between these two. That is how the map over time like has training progresses for each input. For few inputs it just progresses and converges to a optimal map. Similarly your brain does that. Like this is an algorithm for map self-organizing maps, and you have similar self-organization in the brain so that you make sense of the real world. And so this can have dynamic reorganization depending on what input you're getting. For example, if there is an experiment where a cat sees only the world which has only vertical bars around it all the time, and depending on the inputs that it receives over time, the organizing maps only responds to that particular type of input that it receives. So since we are receiving the input from the world, it organizes according to the world. So depending on what your environment is and what uh, in your developmental stages, what environment you're in and how you see it, just the map organizes that way, optimizes that way. This is about self-organizing maps. So I hope the maps is clear, the retinotropic maps and the self-organizing maps, and how like the brain can do some self-organization so that it adapts to the world that it is seeing. Now moving on to subcortical structures, we haven't discussed about auditory cortex, but it has a similar organization like this, with the retinotropic maps having feature space as a frequency rather than a spatial location. Similarly, you have for touch also. You have like neighboring points in your on your skin corresponds to neighboring points, and similar structures, self-organized maps you have for different senses. Now coming to some subcortical structures. So we have discussed about cortex having different lobes. Now going down to subcortex, which has a very important area like basal ganglia and few uh, nuclei, thalamus, hippocampus, etc., which are involved in memory, emotion, primitive, uh, primitive capabilities across all the animals. All those are encoded in this subcortical structures. And this thalamus is what we discussed. It acts like a relay station to many processes in the brain. So it just all the senses come into this thalamus, gets processed and goes back. And similarly, you have motors or signals also coming here. Amygdala is mainly involved in fear conditioning, emotional processing, etc. Basal ganglia is used in like reward-based learning. You have like basal ganglia has certain nuclei which has high dopamine. You might have heard that dopamine is a reward signal, and that way you have these subcortical structures arranged, and these are like a part of basal ganglia, the substantia nigra, and you have ventral tegmental area, etc. So coming to thalamus. So you can see here the thalamic nuclei have different parts going into like projecting onto different parts in the cortex and other subcortical areas or association cortices, etc. So you can see here different functions of different nuclei of thalamus here. This is a bilaterally symmetric structure, and you have different nuclei having different functions, different afferents and efferents. So afferent is that something affecting it, and efferent is this is like affect versus effect. It's the same difference, modulating it and giving some effect. So you have different afferent and efferent connections from the thalamus to different parts of something that is coming to this is called afferent, and going out from there is called effect. Like this affects some new thing, and for example, like visual world affects your LGN, which is in the thalamus, and LGN is again projected onto V1, etc. So this forms the efferent and efferent fibers, and you see different functions and different projections like afferent and efferent fibers from thalamus here. So there is another system called reticular activation system, which coordinates in your sleep, circadian rhythms, various homeostatic processes, homeostasis, to maintain your homeostasis. So it has more diffuse projections. Unlike you saw, like LGN has, for example, LGN has point-to-point connections, point in retina, again projecting onto point in visual cortex, something like that. It has point-to-point connections, whereas this reticular activation system has diffuse projections from thalamus. Like you have this, you have diffuse projection. So thalamus has two types, diffuse and point to point. So this way, like your circadian rhythms are controlled and your various homeostasis stasis processes are there. And it also has like to your movement control systems and then projecting onto your spinal cord. Yeah, this is about reticular activation system and how like two different types of connections that thalamus can have, point to point versus diffuse connections. Point to point we discussed in the, for example, in the sensory processing, especially visual processing. Okay. Uh, so now coming to basal ganglia. Uh, this is a very important subcortical structure which has various functions from reward learning, movement, action selection, etc. So these are like the colored parts on this cortex, so shows the different nuclei or different parts in this basal ganglia. So this part is thalamus. Uh, these parts, these different colors like violet, purple, green, and yellow show few nuclei in the basal ganglia structure. So how is the basal ganglia structured here? So you have two different pathways in basal ganglia, like like cortical connections goes to striatum. So this forms this part, this part forms the basal ganglia structures here. And you have different connections. One is called direct pathway, where uh, the striatum is directly connected to globus pallidus. Here you see this is GP. GP has two parts, GP internal and GP external. And you have different neurotransmitters coming into play at this synapses. So from striatum you have acetylcholine, and you have like GABA projections here. GABA acts like an inhibitory neurotransmitter that helps in controlling the downstream neural activity by inhibiting something like that. And substantia nigra is a uh, dopamine rich nucleus which has dopamine projections onto the striatum. So depending on this levels of reward and cost benefit you have, this dopamine gets like just activates striatum, and based on that your action or different things are selected and then again passed back onto thalamus and back to cortex. This is how basal ganglia is present. So now let's see a model of basal ganglia where it is used in reward processing. So this is also called a race model because you have two actions and there is a race between two actions, some kind of competition, and you have to just select one action based on the cost benefit evaluations. So we have seen these connections, how the basal ganglia is connected. You have like from subthalamic nucleus, you have excitatory connections. This red represents excitatory, whereas blue represents inhibitory. Like these are GABA. And striatum receives input from uh, substantia nigra, which has dopamine inputs, and then it goes back to thalamus and all. And this cortex again affects this pathway. So there is like feed forward and feedback connections coming into play here. So for example, how are two action plans? Let's say you have plan A and plan B. So let's say plan B represented by some nucleus in cortex called PT, whereas A is represented in IT. And what happens is you have two pathways, direct and indirect. So one pathway is for cost, it evaluates the cost, and one path for the benefit. So if the benefit is high, so you see like the amount of blue is high here. So there is a lot of inhibition coming to this GPI nucleus, and since there is a lot of inhibition, this doesn't fire, and there is less inhibition, and the cost minus benefit goes back to the cortex, and you select plan A. Whereas in this, on the other hand, if you're selecting plan B, what happens is there is high excitation to GPI nucleus in this indirect pathway, and since it is excited, the inhibition is also activated to the thalamus, and this cost benefit becomes low, and therefore other action is selected. So this is like some a type of model for basal ganglia here. Oh, so this is how basal ganglia is involved in reward-based learning, where it can analyze cost-benefit ratios, and depending on that, it can select action. And we have seen a model here, and we have seen the structure of basal ganglia, one of the important subcortical area. Coming to hippocampus. So, in hippocampus, which is mainly involved in memory, and especially hippocampus has few cells which are helped in navigating the space, called place cells and grid cells. And so in what happens in place cells is, whenever a rat moves in a maze like this, whenever it encounters a space, like for example a mice goes in a maze where it has rewards at certain points. So at certain point there is a neuron, like one of the place cell always fires when the mice crosses that particular point. This way these are called place cells, whereas grid cells are nothing but like, so a mice is navigating in this some space here. So a particular neuron in a grid cell, like a particular grid cell, fires at different points in the space. That way it has a map of the real world, and different grid cells have response to different spaces, forming like different maps. So that way the mice keeps track, or humans keep track of their own space and relative position and head direction, and depending on that they can navigate the space. So one of the main function of hippocampus is memory and spatial navigation. And you see like different connections in this hippocampus. You have entorhinal cortex, which is a gateway to hippocampus, and then you have CA1 CA3 areas, and then dentate gyrus. So like there are different types of memories, and how the memories are encoded we'll discuss in next lecture. So this is about how the hippocampus is like basically the function of hippocampus. So now coming to different types of uh, coding, the different types of how the neurons encode information of real world. There is a type of coding called selective coding. What I mean by selective coding is, for every object you have a particular neuron that is responding. Like you have a set of three neurons, and each neuron responds to one particular person here, or one particular object per se. So this way, like your neurons are able to identify which person you're seeing or which person you're interacting with. But this seems to be inefficient, because like it requires every neuron to represent every object, and it just uh, the number of neurons required just goes very high. So a better version of representation is something called population coding, where uh, the information for particular object is represented in a population of neurons. It's also called distributed coding. So distributed across three all the neurons in particular population. So you have a different persons have different types of responses in three neurons. Like neuron 1 has a response, neuron 2 has, like this is like some kind of binary coding you can do, 0 1 1, something like this. So this way, this is another type of coding where your brain can represent the information, the neurons in the brain. Here we are just taking example of three neurons, but in fact it is a very complex space representing the different objects. So and there is another type of coding called sparse coding, where in a population, like here you have three neurons, but you can have a uh, cortical structure of 10 neurons here, and only few are activated for particular object and differently activated. That way you have uh, you know less amount of uh, resources could uh, inform could encode for the real world objects or people or any information in the real world. So sparse coding is a type of population coding where only very few number of neurons are active from the population. Instead of all the neurons getting activated differently, few neurons are activated and those few are differently activated. That is about sparse distributed coding. The three different types of coding apart from the coding that were discussed in the lectures, where you have like temporal binding and combination coding. So what I mean by 1 second, yeah. So what I mean by temporal binding is you have an object which is like yellow triangle and blue square. So there are different set of neurons processing this color and processing this shape. But how does one know, like how does your brain know that it is an yellow square or blue triangle or blue triangle or blue square? So there should be some kind of combining the information for representing a particular object with multiple features. So the neuron population representing this neuron and this neuron could fire synchronously, like they have same timing when they fire, and this way they're able to combine the information and send it to downstream, and like this neuron synchronously firing can send it to downstream neuron, and this can make sense of the object, whether it is yellow triangle or blue square. Whereas combination coding is more like an hierarchical processing, where you have primitive, like simple features hierarchically being processed and combined, and then there is a neuron which responds only to particular combination of features, and that way it makes like that way it encodes whether it's a yellow triangle or blue triangle. So in brain it has like there are different types of coding possible. There is not one specific coding that is called optimal. So it is a mix and match of various things, and then uh, since it's a complex device and depending on what kind of input it receives, it can combine in different ways. So this is about it. So yeah, any questions, any further questions we can discuss from the lectures or here. If not, you can ping me on the discussion forums. This is about week 3 lectures. There are a few more things like the phantom hand experiment, etc. That is a kind of binding two features from like visual features and sensory features, and you have discussed like that was discussed in the lectures. So okay, okay then. See you guys.